Hello, class. So, last time we talked about graded potentials and resting membrane potentials of neurons, and all of those action will result, well, that is, if there is a stimulus that is great enough, into an action potential. So, action potentials. What are action potentials? So, the action potential will occur in a neuron when a stimuli occurs. So let's look at this step by step. So the main idea here is, let's say a neuron is stimulated. So stimulation occurs, right? Let's say a stimulation has occurred. And obviously, with the stimulation of the neuron, this will result into the gated ion channels opening. So gated ion channels open, right? So they will open. After they open, this will generally generate the graded potential. So the graded potential occurs, and either it can go through hyperpolarization, in which in hyperpolarization the membrane potential goes below the resting membrane, as we've learned prior, or it goes in depolarization, in which it reaches above negative 70 millivolts. So it's going higher. And this will generate an action potential if the threshold is met. So that's just a recap of what happened, or what we discussed last class. I want to talk about how action potentials work, because we've already mentioned how graded potentials work. We're going to look at how action potentials work step by step. I'm just giving a recap to kind of prepare us in understanding how action potentials work, because generally the way an action potential work is that if there is either a graded potential that will summate, right? So graded potentials that will combine and reach the threshold, or if there is an a stimulus that is strong enough and it just triggers an action potential. It just reaches threshold right away. So just want to recap that. So an action potential occur either by graded, let's say, summation, right? Graded summation, or a very strong stimulus. Let's just say you touch something really hot, boom, very strong stimulus. Or your pain receptors on your nociceptors are stimulated. Okay, so let us look at action potentials step by step. So, action potentials. So we are going to need this graph in order to better understand how these action potentials work. So some things to remember is that action potentials will have trigger zones. These trigger zones will be, let's say, on the axon hillock, right? So this occurs. So for multipolar neurons and bipolar neurons, this will happen at the axon hillock. Okay? That's where you will have these action potentials occurring. In the unipolar neuron, you will have this happening at the dendrite. Okay, so you will have action potentials being initiated at trigger zones at the dendrites of unipolar neurons. And you will have action potentials being triggered at the multipolar bipolar neurons under axon hillock. So on the axon hillocks of these types of neurons and on the dendrites of unipolar neurons. Okay, let's look at this step by step. So let's say a stimulus has occurred at the neuron. So this is a resting membrane potential. Actually, let's switch that here. Okay, so that is our resting membrane potential. And let's say there was a stimulus that was great enough to trigger it to over here. Let's say this is negative 55 millivolts, which is our threshold in initiating. Okay, so step 1. Let's say a stimulus happens on the neuron. Okay, so this is because of a graded potential summation, so multiple instances resulting into meeting the threshold, or just one big powerful stimulus. So stimulus on the neuron by summation, reaching threshold, or this is by powerful stimuli. Okay, by one powerful stimuli. Now that there is a stimulus on the neuron, we go into the next step, which is step 2. Depolarization will occur. Now that you have reached the threshold, okay, so step 2 over here, depolarization will take place. So the membrane potential of the neuron becomes more positive. So it's going to go up, 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 and away. So it's going to go increase, increase, increase. Okay, so that will increase until we reach the peak potential, which is around 30 millivolts over here. Let me just put that over there. So depolarization will occur. Okay, as you can see here. Now that because we've reached the threshold to generate the action potential. So when depolarization occurs, this will happen at negative 55 millivolts. That's a very important value to remember. This will occur because enough sodium voltage, not just your regular leakage channels, but your voltage gated channels will open. Okay, so enough of these have opened. This will obviously result into the increase in sodium permeability. With the increase in sodium permeability, as we have learned in the previous lectures, this will make the neuron much more positive. Okay, so the inside becomes more positive. Now that the inside becomes more positive, eventually the neuron's membrane potential will reach what I would call the peak potential. So that is what is happening here. So this will occur at 30 millivolts, positive 30 millivolts. So step 3. So peak potential occurs. Okay? Peak potential. That's just my terminology. Peak potential. So the peak is reached. This occur at 30 millivolts. Positive 30 millivolts, not negative. Positive 30 millivolts. And when we reach the peak of this action potential, the sodium voltage gated channels will inactivate. So sodium voltage gated channels inactivate. So they will inactivate. Once we reach that very peak of this action potential, they will close. Okay, so they will close. This will result into then repolarization. So you're gonna start seeing this decrease. Okay, so it's gonna decrease. So step 4, you are going to see repolarization. In repolarization, now that the sodium voltage gated channels have closed, you have the potassium gated channels opening very slowly. And as they open very slowly, you're going to see the neuron becoming negative again, as these potassium ions, as we know in a resting membrane potential, they will leave the neuron, making the neuron more negative. Because remember, the potassium are positive charges. Now that we have these positive potassium ions just leaving the neuron becomes more negative again, as we can see here in repolarization. So the potassium voltage gated channels will open slowly. Obviously, this will cause an increase in permeability in potassium. Sorry, I spelled that wrong. There is an increase in permeability in potassium. You know, we have more potassium voltage gated channels open. Now that repolarization has taken place, this will also then result into hyperpolarization. So it's actually going to go a little bit below the resting membrane potential of negative 70 millivolts. So that would be step 5. So hyperpolarization. So hyperpolarization takes place. The membrane potential of the neuron becomes more negative. Than the resting membrane potential of negative 70 millivolts. So let's say this will be at negative 75. The neuron has become more negative. This is because the potassium voltage gated channels not only open slowly, but they will also close slowly. Okay, so the potassium voltage gated channels will close slowly. We then return into the resting state at negative 70 millivolts. So step 6. So resting is then reached again. When the resting membrane potential is then reached, the amount of potassium going in and the sodium going out will equal. Okay? Or in other words, the amount of sodium going in equals to the amount of potassium. Sorry about that again. The amount of potassium going in will equal to the amount of sodium leaving. Remember the sodium-potassium ATPase, right? Where we have 2 potassium coming in and 3 sodium leaving. Here, once we reach that negative 70 millivolts again, we would actually have this state where we have the amount of potassium going in equalizing the amount of sodium leaving. Okay? Some things to remember. While the action potential is occurring, we have the sodium-potassium ATPase working, okay? This doesn't stop working just because there is an action potential. This will continue to work to maintain the gradient. And then it will generally take around 10,000 action potentials, 10,000, to see a measurable change in ion of the cell. Okay? Okay, so we discussed how the action potential works. The question is, why would the action potential even happen? Remember, action potentials occur in order, so they allow communication between different neurons. So they act across the entire length of an axon, so allow communication. Okay? So they allow communication. Think of them as like electrical synapses. Okay? They go through the entire length of the axon. Okay? And the thing with action potential is that if the threshold is met, so not only that they allow communication, they also happen or don't happen. So remember that value of negative 55 millivolts? If this is reached, an action potential occurs. If this is not reached, no AP if it is not reached. Okay? That is what we would refer to as the all or none principle. So the action potential allow communication by propagating along the entire length of the axon, while the way that these action potentials work is that either they happen by reaching the threshold or they don't happen if they don't reach the threshold. So unlike the graded potential where it just happens because there's a stimulation, here either it happens or it doesn't. Now that these action potentials allow for communication, the way that these charges will work is that they will actually go in both directions. So as you can see here in this diagram, the charges will go in both directions of the axon. However, the action potential, so the action potential itself only occur in one direction. Okay? The reason for this is that this side where the action potential has not occurred or have already occurred is in what we would refer to as an absolute refractory period. So over here, let's say the action potential is happening here, okay? So AP over here. And let's say there was already an AP action potential here. This area would be an absolute refractory period, meaning, I'm just gonna put meaning that no matter how strong the stimulus is, you're not gonna have those sodium voltage gated channels open. And we are going to look at that. The other thing is that not only that they could be in absolute refractory period, let's say this was the trigger zone of the dendrite of a unipolar neuron. Before the dendrite of a unipolar neuron, the structure to trigger an action potential is not possible to allow that action potential. Remember the trigger zones of the action potentials we mentioned in the beginning of the video. For unipolar neurons, it's dendrites. For multipolar neurons and bipolar neurons, it's the axon hillock. So if it was triggered there, obviously you're not going to have an action potential occurring prior to those trigger zones because you want the signal. Remember, action potentials are like signals. You want it to go in one direction. So either they are, in absolute refractory period, or they have a different structure altogether. So you have charges going both directions, but action potential in one direction. Okay, so let us look at refractory periods. The first one that we are going to look at is the absolute refractory period. In the absolute refractory period, no action potential can be generated regardless of the size of the stimulus. Now, this is because either let's say all the sodium voltage gated channels have open, okay? Let's say they are all open, and this generally occurs when we have the depolarization, right? And it's reaching to that peak potential. All that sodium voltage gated channels are open, and there's no more that we can open. That is an absolute refractory period. Or the other factor is that the sodium channels are inactivated and they cannot be activated again until the membrane potential reaches the resting, which is -70. So either A, all sodium voltage gated channels are open, or they are inactivated. And this is very common occurrence, as we can see. So we have our depolarization, our peak potential, and now we have our repolarization. So this will occur, let me just put this here, at repolarization. The other refractory period that can happen, so if absolute refractory occurs at depolarization or at repolarization, we also have relative refractory. Okay, so for relative refractory, an action potential can be generated only if that stimulus is greater than normal. So, AP for action potential can occur if stimuli is greater than normal. And this will generally occur at hyperpolarization. So, here is a graph. So we mentioned the polarization, the peak potential, hyperpolarization, and now here, so depolarization, right? Peak, and then repolarization, now the hyperpolarization. So it has to be, that stimuli has to be greater than normal, because look at this, it is below the resting membrane potential. So obviously you need that stimuli to be really great. Now, at least there could be an action potential here, unlike in the absolute, where no matter how strong the stimuli is, you just can't have one. But here, you can have an action potential. It's just that the stimuli has to be very strong and has to be way beyond normal. Okay, so you're gonna need a larger stimulus because it is farther to reach that threshold. So if, let's say, negative 55 is here, look, this was the resting over here. Now you're all the way down here. So you're gonna need a larger stimuli. Does that make sense? I hope that makes sense. Okay, the last thing we are going to touch today for this class is the rate of propagation and how this will affect our action potential. So the rate of propagation will depend on our first factor, being the fiber. Now, if the axon diameter is larger, right? So let's say, there we go, fiber axon. So obviously larger diameters will result into faster propagation compared to a smaller one, because obviously if it's smaller, there's more resistance. You're going to have slower propagation of that action potential. The other factor that influence the propagation of our action potential would be myelination. Now, you can think of myelination like electrical insulation, okay? So if we don't have this insulation, for better current flow, you will have slower propagation compared to a neuron that has this myelin, which will result to faster propagation. So let's say if you have an unmyelinated you're going to have action potentials that are going to be a lot slower, okay? Basically, you have the sodium channels that are opening adjacent to each other. So let's say this is, we're going to draw a neuron here. So let me just find the proper pen. So let's say here is a neuron, cell body, dendrites, right? Here is our axon. That's a very interesting neuron. Okay, so what you're gonna see, remember that opening of that sodium, it's gonna open adjacent. Okay, this is going to go in what we would refer to as continuous conduction. So in an unmyelinated neuron, we have a very slow propagation. Okay? The propagation is very slow. Now, if it is myelinated, we are going to have a faster propagation. Sorry, I spelled it wrong. That's one I. Myelinated. So you are going to go through saltatory conduction. And we're going to look at that. So let's say here's your neuron again, cell body. Here's your axons. Now you're going to have these nodes of Ranvier over here. And let's say over here. There's your terminal. Okay. So you're going to have not going through adjacent. Okay, so the sodium are not going to open adjacent like here with the unmyelinated. Instead, let's say this is sodium. It's only going to open those sodium voltage gated channels at the nodes of Ranvier. And this will go through saltatory conduction. Basically, it's going to propagate a lot faster. The way I would think of this is, let's say you are in a road trip. Okay, so here is a car. Not the best car, but here is a car. Now, here you have to basically open the sodium channels every adjacent of that axon. So what this means is that the car gets refueled often. Okay? And obviously, if the car needs to stop and refuel and stop and refuel and stop and refuel, you're going to travel a lot slower. That's the same idea here. The sodium opens every adjacent of this axon, while the propagation will be a lot slower. Compared to, let's say, over here, the sodium channels only open at the nodes of Ranvier. So it's only going to open every once in a while. Here, back to our analogy, the car gets refueled less often, resulting to a faster travel. Okay? Let's say this is the map of Canada. Here's Quebec. Not the best. Manitoba. Etc. etc. If you have to refuel every bit, you have to stop every bit, you're gonna travel a lot slower compared to refueling here, refueling here, refueling here. Okay? So here with the unmyelinated, it is a lot slower. With the myelinated, we have a faster propagation, resulting into saltatory conduction. Now, there are also different types of fibers that is affecting our propagation of our action potential. This will be our last topic for today. So, we have what we would refer to as the type A, and then we would have our type B. Remember, earlier we mentioned that fibers affect propagation and obviously myelination. these fibers will obviously affect our propagation. Now, which one do you think will have a larger diameter? It would be type A. So type A would have a large diameter. And if you have a large diameter, as we mentioned, we are going to have less resistance, okay? And this will generally propagate around 100 or 130 meters per second. And this is found in most sensory neurons, motor neurons, and skeletal muscles. I'm just gonna put SKM. You are going to mostly find these types of fibers in sensory neurons, motor neurons, and skeletal muscle. The reason being is that you want really fast, okay? So I will just put 130 meters per second here. You want a really fast propagation. Let's say you touch something with your feet and it hurts. You don't want a very slow propagation of those signals because it's painful, right? So you want that skeletal muscle to synapse onto the neuron very quickly. Okay? You want that synapse between the neurons, so as it goes from the skeletal muscle onto the neurons, you want it to travel very fast. You don't want it to be slow, or else you're gonna be reacting to everything slow when you touch something painful. You then have the not type B. You are going to have the type C fiber. The type C fiber is basically the opposite of type A. You're gonna have, smaller diameter. You're going to I almost forgot. In type A, it is myelinated. Okay, so this is myelinated compared to this one in type C, which is unmyelinated. We already mentioned how myelination works. I hope that would be a lot clearer to understand. So yeah, it is the opposite of type A. Type C has smaller diameter. It is unmyelinated. The propagation is slower. You're going to have propagation of around 0.5 meters per second rather than 130 meters per second. And rather than being found in the skeletal muscles, sensory neurons, and motor neurons, you're going to find these type C fibers in autonomic areas and some pain fibers. So the autonomic nervous system and some pain fibers. Okay, so I hope that helps clear everything for today. So today we've learned how the action potentials work on a step-by-step process. We mentioned how there are factors affecting the propagation of that action potential. We've looked at the fibers, the idea of myelination, how they travel between unmyelinated versus myelinated, and we've looked at other influences such as refractory periods on the different phases of action potentials, and we've also looked at other important information of how these action potentials work, on why they only go in one direction, not in two, and how charges actually go in both ways. Okay, I hope everyone has a good day. Thank you for joining for today's lecture. Our next topic will be on synaptic transmission across the synaptic cleft.